Hello, everyone, and welcome to this special lecture for Paleocast. I am Dr. Alejandro Izquierdo-Lopez, and this will be the third part of this series of lectures. In the first lecture, we learned about what is the Cambrian Explosion, what happened 500 million years ago, and what does this have to do with the early evolution of animals. In the second lecture, we discovered how was the Cambrian period, what type of temperature, what type of ecosystems did it have, and where is the Burgess Shale located, and why is it important to study this unique fossil site. So now I'm going to go and merge both of these lectures together and basically talk about the early evolution of animals seen from the latest Burgess Shale research. What I'm going to do is go through every single group of animals and see how the Burgess Shale gives us new information about how they first evolved. So let's start with a graphic about all the animal phyla. So what is an animal phylum? Animals can be classified into different groups like classes or orders, and one of the largest ways to classify animals, perhaps not the largest, but one of the largest, is the phyla. The phyla are also called body plans or body organizations, and basically relate that this group of animals had like a specific number of characteristics that are all shared by this group of animals. So, for example, we will have like vertebrates here, like cats and fishes and other types of animals. We will have, for example, a backbone. That will be one of the conditions to belong to the animal phyla called vertebrata or chordata. So, this is probably one of the most comprehensive trees of animal evolution. They are on the internet. This is made by myself. And I try to put almost all the main phyla that we can think of. However, I have to say also that some of these relationships may be disputed by some different authors, and also that maybe some groups may not be constituting one phyla, maybe part of another phyla. For example, one of the most common ones will be around the natifferents here, where we have the natostomulids and rotiferates and also on. So maybe these groups just are part of just one big phylum. Because at the end of the day, these classifications are human-made. So there's a lot of like nuisance on how do we classify them and whether we consider them one phylum or we consider them like two or more. So as you can see, this is similar to the graphs that we have seen before, where most of these animal groups originate before the Cambrian period, mostly during the Acheran period. But of course, I would like to remind you that it is during the Cambrian period that we have their first fossil record of these animals, at least of most of them. As I mentioned previously, there are some instances of potential sponges, jellyfishes, as well as mollusks, and I think um, annelids are also potentially present in the diagram, but even those fossils are not super convincing. So I will go through every single part of this tree and just talk about it. So the first part will be the basal groups. You see, evolution of the animals will start here, and those will be the earliest branching groups of animals. So we can consider them as some of the most, let's call them primitive, but of course they have been evolving the same time as other groups. It's just that they have retained many of the characteristics that probably were present in the common ancestor. So, what are the basal groups of animals? We have different ones, and I will start with porifera, which are sponges. So, poriferans appear also like very early, and there are probably like some maybe fossil record from the Diacaran. Of course, there is clear fossil record from the Cambrian. And I'm gonna talk about how do we identify a sponge? Well, how do we identify every single animal group and how do we identify them at the Burgess Shale? So, what are the traits that characterize this phylum, and also how can we see that at the Burgess Shale? That's because many of the traits that characterize, for example, sponges, are not seen on fossils because they are part of their cellular structure, for example. So, when we see a fossil at the Burgess Shale, how do we know it's a sponge? Probably because it has spicules. So spicules are the skeleton that makes the body of most sponges. There are some sponges that may not have exactly spicules; they have other type of structures, but spicules are mostly found in the majority of them. We'll have an amorphous, non-symmetrical body that would indicate that it's not another more, let's call it more advanced group. And also we will have some type of pores and canals. So that's how nowadays sponges look like. No, we have this structure where we have a body made out of like pores and canals, and all of this structure is made out of like muscles as well as the spicules and different types of cells, not many, like 4 or 5 types of cells. And then what sponges do basically is they get water throughout their pores, and then they expel it through their main pore here at the top. And while doing that, they just filter the water and they capture organic particles. And there are, of course, some different sponges. There are carnivorous sponges, but we do not have evidence of that in the Cambrian. So, how many sponges do we have at the Burgess Shale? We have around 22 species. You can find them all here. So we have quite a good diversity of sponges. And what have we learned from the sponges at the Burgess Shale? So here we have another one. So they were not all of them, sorry. Here, that's what have we learned about the sponges from the Burgess Shale. So we found, for example, different ecologies that we do not find nowadays among sponges. For example, some of these sponges, like choya, were type of like they were floating like through spicules. So they had like these very very long spicules, and they basically were used these spicules to basically be on top of the ground, and that's a very strange structure that is not found nowadays. But you can also see other types of sponges at the Burgess Shale. For example, Petaloption has clearly the pores, so that's how it's classified as a sponge. And then we have, for example, piranha. Piranha is an interesting sponge because it has brachiopods, which is another animal that has a shell, and this animal is stuck on top of the sponge. So we think that maybe they were in symbiosis, or maybe these brachiopods were commensalists, so they were taking advantage of the filter currents of the sponges, or maybe for habitat or protection. But it's an interesting association between two groups of animals. But we also learned a lot about the origin of these spicules, the skeleton sponges. These are, for example, the diversity of spicules that we find nowadays among sponges. You can see very, very different types of spicules of different shapes, number of points, and so on. But in the Cambrian, we have very unique spicules that were not present before, so that were not present now exactly. So we have, for example, diagonella. Diagonella is a sponge where you have this kind of like stitch patch type of spicules, which are very interesting because that's nothing like we see nowadays. So there was some type of spicules that at some point just went extinct. And then we also have the spicules of aphelia. These are very interesting spicules because nowadays most spicules can be classified as carbonaceous or siliceous. So they're made out of calcium carbonate, or they are made out of silicon. But in euphilia, we have basically a core made out of silicon and then an outer layer of calcium. So some people have suggested, some of these authors here, that this was probably a sponge that was the ancestor of the two groups of the sponges that were ultimately doing spicules of one kind versus the other kind. And this was the ancestor that could do kind of a mixture of both kinds of spicules. But this is not the only thing. We have, for example, Takakawa, which is this very strange sponge and has this very unique type of spicules that are kind of like serrate. They have like kind of like teeth-like shape here. And we even have something very, very simple sponges, like these X-like sponges that are from Protospongia, which is considered perhaps one of the earliest type of sponges. And that's because they will have these very simple spines of like X, and they are not structures in kind of like a mesh. They're very placed kind of like randomly. So we have overall a very wide array of different types of spicules. So these sponges, and now let's talk about ctenophores. Ctenophores are called comb jellies. And comb jellies, again, they have a fossil record that starts at the Cambrian period. I don't think we have good evidence of previous ctenophores, and they're probably some of the most ancestral animals. So, how do ctenophores look like? So, ctenophores are not jellyfishes. They lack some things that jellyfish have. For example, many ctenophores cannot, or the majority of them do not cannot stink, so they don't have the cells that jellyfish have that they are used to stink, and instead they have this row of cilia that are like small hairs that, for example, underwater they can create some type of like light diffractions, and that's why they in the photos they always have like these colorful patterns. So how do we identify a jellyfish, well, a comb jellyfish in the Burgess Shale? We understand that the common jellyfish should have a radial body, like a jellyfish. So we'll have a radial body basically means that from the top it will look like round. We will have the series of cilia, so the combs, the comb jellyfish combs, and maybe we have some tentacles. So comb jellyfish sometimes, not always, have tentacles, but not as many as the normal jellyfishes. We have this corona. No, if you picture a jellyfish, you'll find a lot of different tentacles here, but comb jellyfish normally have like maybe like a pair that are very long. But of course, there is some diversity. So, what are the ctenophores of the Burgess Shale? We have only three species, so fewer than poriferans, for example. And what have we learned from them? Well, we learned that they look mostly like we expect them to look right now. So they haven't changed that much in 500 million years. We have, for example, Chanioascus. And Chanioascus, you can see here these black patterns, which can be identified as the row of combs of the comb jellyfish. But for example, Chanioascus is also very interesting because it has these globular structures here, and you can see them pictured in the reconstruction by Marian Collins, but we have no idea what they are. We don't have any structure on comb jellyfishes that look like this nowadays. Maybe it's some type of organ, maybe some type of, we don't know. But we clearly see them inside the fossil. So ctenophores, again, we only have three species, and they do not provide much information about their early evolution. Maybe the main take-home message is that they look almost exactly as they look nowadays. Let's go now for true jellyfishes, for cnidarians. Of course, when I talk about cnidarians, I include jellyfishes, anemones, and corals all together. So, of course, like nowadays, the easiest way to identify jellyfish is by looking at its cellular structure and identifying these very small cells that have like a harpoon, and that's what they use to sting. However, that's not something we can see inside the fossils. So we have to rely on the radial body. We have to rely on the presence of tentacles, which may not happen because sometimes they are not preserved or sometimes they are not there. And yeah, we other types of traits are not present. So having a jellyfish during the Cambrian period fossilized and being convinced that this is a jellyfish is very difficult. So that has happened in the last years, in the last, well, since the Burgess Shale was discovered, we have a lot of different types of jellyfishes, and in the last years there have been some uh, pretty convincing evidence about these organisms being jellyfishes, but they still look very strange. So we have, for example, McKinsey. McKinsey is one of the largest animals of the Burgess Shale. It can grow, I think, up to 30 or even more, 40 centimeters. So it's quite a very long organism, and it doesn't look anything like a jellyfish. But it kind of looks like an anemone, so that's what has been classified as. And then we also have Thaumatylon. Thaumatylon is this very strange jellyfish that looks also like anemones nowadays, like some of them that are like called like sea plumes or sea feathers, and they have like a similar structure, and that's why they have been called jellyfishes. But again, they are very strange. They're kind of potentially more anemone-like, but very unique organisms. But this all changed until this year, where one of our previous PhD students, Justin Moon, basically described this new jellyfish. This jellyfish had been known for a while. It was part of one master thesis and was discovered in the Raymond Quarry around the 80s. But no one had taken the time to actually look at it. So Once both Jaman Arcaron and Justin Moon look at this jellyfish, they conclude that was probably the most convincing jellyfish of the Cambrian period. And you understand that this will be also not an anemone, it will be clear a free living medusa. So it will be a jellyfish that was moving and was very similar to what we find nowadays. So this is classified as, again, probably the earliest jellyfish in the fossil record. We have other potential jellyfishes, but this is the most convincing one. You can even see the tentacles here. And there are also like some organs inside the jellyfish, which we don't know what they are. Maybe they are gonads, so reproductive organs, but it's not clear. So we have these basal groups, and then what about other groups that are very basal? Unfortunately, we don't have a good evidence of these groups being present at the Cambrian or at the Burgess Shale. So there's no evidence of chinoscelomorphs, of placozoans, and there's other places that are outside of the Burgess Shale that preserve archaeocyathids. Archaeocyathids are a group of organisms that look kind of like sponges, but they are different because they have kind of like a tabulated type of body. So these are different from sponges, but probably closely related to them. But they are not present on the Burgess Shale; they're present at other sites. So we go back to our main tree, and now let's look for another group. Now we're going to look at the deuterostomes. So when animals first became bilateral, so they acquired bilateral symmetry here, we have two main groups that basically are differentiated on how their embryo grows. And of course, we have the protostomes here and the deuterostomes, and that's where we are eventually, the deuterostomes. So the deuterostomes nowadays, as I said, they are mostly identifiable for how their embryo works and also how their anus and mouth are formed. Of course, that there's of course like genetic evidence for this group that they are closely related and includes things from starfish, hemichordates, some strange animals called the betulitolians, urochordates, sometimes called sea potatoes, and us, so chordates and other type of vertebrates. I use chordates and vertebrates differentially because basically, if you are really into like vertebrate anatomy, you will make probably a clear difference between what is a chordate and what is a vertebrate. But most people are not that familiar with the word chordate. That's why I use vertebrates. But I acknowledge that this is not exactly the same. So there's some evidence of like potential deuterostomes, of maybe early deuterostomes that were before they branched into different groups. For example, this one here, which is Eldonia. The name is not written, but Eldonia is kind of like a disc-like animal with this type of like structure, and this is believed to be one of the earliest deuterostomes. But again, it's a very strange fossil. It's very difficult to classify, so it's no wonder that some people may have different opinions on what does this animal look like. Where does it place in the animal tree? So now let's go for more familiar organisms. Let's go to echinoderms, which Echinodermata has sea urchins, sea lilies, starfish, and so on. Echinodermata. So echinodermata are quite easy to recognize in fossils and at the Burgess Shale, mainly because some of the characteristics that typify them can also be easily fossilized. One of them is that most echinoderms, not all of them, have some type of pentaradial symmetry. So you can see like it's like star shape. You can also see if you have like a fossil sea urchin, you will also have this pentaradial symmetry. If you cut the stem of a sea lily, you will also see this pentaradial symmetry. The other thing is that it has also this very unique exoskeleton made out of ossicles. So it's made out of very small plates that cover all the body and they have other structures. And finally, what identifies them is that most echinoderms move and feed thanks to a water vascular system. What they do is they capture water and they bring it through these tubes, and basically using water as a way to move and as a way to transport particles and transport food. So water is kind of, instead of having us like blood, we have they have also like water. They also have blood, but they have water mostly. Well, water for this function. Or they have blood for other functions. And then we have different types of echinoderms at the Burgess Shale. We have some of them more convincing than other ones, and I'm gonna go a little bit through them. So there's not much diversity. There's around four or five I've used seen. One, for example, of the most amazing echinoderms is Lyrasastis or Lyrasistis. And Lyrasistis is what is believed to be an eocrinoid, so probably one ancestor to nowadays sea lilies, so crinoids. So these animals live nowadays in our seas, and probably Lyrasastis was an ancestral type of of crinoid, so called eocrinoids. Um, we also have, for example, a very interesting group called the Adrioasteroids. The Adrioasteroids are a group that's found all across the Paleozoic, and they have this kind of like shape of like a. They look like a coin, and in the coin you can see this pentaradial symmetry pattern. No, they have like this kind of like structure that is pentaradial, and they look like kind of like again like coins. And we have, for example, at the Burgess Shale, we have Walcotidiscus. So echinoderms, we have some of the earliest evidence of the radiation of the first groups of echinoderms at the Burgess Shale, but we don't have much evidence of very unique novelties. We do have, though, which I have not talked about, some strange things that we are not sure whether they are crinoids or not. For example, Ecmatocrinus, we think that's a putative echinoderm, but again, it's still quite strange. Let's talk now about hemichordates. Hemichordates is a group nowadays that is also found in our seas, and we have two types of hemichordates mostly. We have the hemichordates that kind of like do a tube, and they create like some type of like filtering structures, and then we have the hemichordates that live just like under the mud, and they basically have this three-segmented structure of a proboscis, a collar, and a trunk. And they use basically the proboscis to make holes, and then they use the collar to filter particles, and so on. So these are the pterobranchs and enteromneus, in case if you are interested. But that's the main two groups of hemichordates. So how do we identify them? It's also more or less easy. So if it's an enteroneus, it should have the three-segmented body structure of the proboscis, the collar, and the trunk. And if it's a pterobranch, you should have some type of tube, and then on the tube we'll have some type of filtering structure. But of course, we have a problem with pterobranchs, which is that many other animals do the same. For example, polychaetes, annelids, there are some type of annelids that also do a tube, and then they create some structure like that. So it's not may not be as evident. What type of hemichordates we have at the Burgess Shale? So we have, for example, Caunograptus, Giles englossus, Oisia, and Spartobranchus. And I'm going to talk a little bit about the research because it has been highly developed in the last years. So for a long time, we had some type of organism at the Burgess Shale called Margaritia. And Margaritia is this structure here, and it was described as a green algae. But then everything changed, and we now think that it's not a green algae. So what it is? So Carmen Anglo, previous PhD student from our lab in 2016, described fossils of Oisia, which is this hemichordate, and this hemichordate was inside this Margaritia. So we think that this is the same fossil. It's just that this is like the tube, and this was the organism living inside. And you can see here the reconstruction. So the tube, the Margaritia, and here the Oisia. So you can see already something interesting here, which is we don't have the typical structure of one pterobranch here inside its tube and creating the filtering structure. We have kind of like a mix between both. No, we have the animal with the three-segmented part, and we have the tube. So that's already pretty interesting. And this is further emphasized by the idea of Gaia Senglossus, also described by Carmen Anglo. And this is a very strange organism because, again, it's kind of a mix between two. We have the three segmented structure, but we also have some type of filtering structure. So all of this tells us that perhaps during the Cambrian period, we still don't have a full differentiation between these two groups, between the therobrines and the pteronodes, so between the tube-feeding animals and the free-living animals. But instead, we had some type of ancestors that were doing both combinations and then later maybe specialized into different, to specific groups. And we have that, for example, in the Gaialsenglossus and Oizia. And then we have other strange things. For example, Herpetogaster. Is Herpetogaster a hemichordate? Well, it does have the filtering structure, but it's not inside the tube. It kind of looks strange. Has also been classified as an early deuterostome. So we don't know exactly what is Herpetogaster, but we know that it's probably a deuterostome. No, it's in this group. So now we go to Betulicolids. Betulicolids is a very strange group that is not living nowadays. So this group at some point went extinct, but we do have some fossil record. Betulicolids are basically recognizable because they have some type of two-segmented body with a tail-like structure and some kind of like funnel-shaped body. And then they also have some type of five oval structures normally here that they are interpreted as gills. So that's why we think they are probably very related to hemichordates and chordates because all of these groups have gills. No, they have these kind of like slits into their throat that they use to breathe and also capture particles. So betulicolids have these gills, have two-segmented body, but they are still very strange. We only have one betulicolid at the Burgess Shale, which is Bamphian.